Okay, I think we'll get started since it's after the hour. So it's a pleasure to introduce today's speaker, Yanlin He. Dr. He is at Baylor College of Medicine, where he's in the Department of Pediatrics and the Children's Nutrition Research Center at Baylor College of Medicine in Houston. He did his undergraduate work in biomedicine at Nanshan University in Shaanxi, China. And from there, he went on to do his Ph.D. in biophysics at Fudan University in Shanghai, in China as well, of course. And then he came to the United States initially to do his first postdoc at the University of Connecticut Medical Center campus in Farmington, where he worked in the cardiology center there. And from there moved to Houston to join the group he's presently at as a postdoctoral research fellow at Baylor College of Medicine. Dr. He told me some interesting stories about his personal career and ending up in Connecticut and working in some very interesting areas in electrophysiology of cardiomyocytes, yes. And then really rejoining his primary interest in neuroscience with his move to Baylor College of Medicine. His primary interest is in central nervous system regulation of glucose metabolism and eating behaviors, and how misregulation here can lead to diabetes and obesity. He is a PI right now on an American Diabetes Association fellowship, where he's studying the effects of estrogen on neuronal responses to hypoglycemic events in diabetics. And he's published quite a series of publications. He has 28 publications, of which he is first author on most, certainly many of those, and they're in terrific journals, Nature Medicine, Nature Neuroscience, Nature Communications, Cell Reports, Biological Psychiatry, Journal of Clinical Investigation, Journal of Clinical Investigation Insight, etc. And I should add, a number of those Nature publications and Cell Reports papers, he has multiple publications in each of those journals. So his work continues to receive very high visibility coverage for the impact that it's having. I'll just briefly mention a couple of things he's been doing recently that I think are germane to the discussion today. For example, his most recent Nature Medicine paper looked at Asprosin, which is a newly discovered fasting hormone that's involved in hepatic glucose production. And it's very interesting in that they showed it not only gets through the blood-brain barrier, but it acts on orexigenic neurons in the brain and works through, they showed, a cyclic AMP signaling pathway. And through that can actually stimulate appetite and have an effect on body weight and obesity. So really getting at the cellular and underlying circuitry mechanisms that lead to behavior, and in this case, obesity. His most recent Nature Neuroscience paper, they looked at nuclear receptor co-repressors regulating cognition. And in particular, we're studying regulation of GABA receptors, GABA-A receptors, and lateral hypothalamic neurons of the hypothalamus, and how that downstream can modulate cognitive events such as learning and memory and LTP in the hippocampus. And then lastly, I'll just mention their most recent Cell Reports paper, where Yanlin has discovered looking at interaction and role of 5-HT in modulating dopamine pathways. And through a very elegant combination of neuroanatomical tracing, electrophysiology, and behavior, was able to really work out in much more detail and understand the neural circuit that's involved with inhibition of binge eating in the brain, and the connectivity between the dorsal raphe serotonergic system and the VTA dopaminergic system, which I think has even greater implications for other areas of cognition. So without further ado, welcome, and look forward to your talk, Yanlin.
All right.
Sure, your microphone is turned on.
Okay, all right. Thank you, Dr. Fralin. Thanks for your introduction. Hi, everyone. My name is Yanlin He. I'm from Houston, Texas, a city that easily sets you on fire during summer. And the reason I'm using this film name is that I think this best summarizes the AgRP neuron activity in response to hunger. The reason I'm very interested in this group of neurons is the people found that it's very essential for feeding. And this group of neurons co-release AgRP-related peptide and neuron peptide 1 and also GABA. But in the rest of my talk, I'm only call it AgRP neurons. You will see here in this picture, the AgRP neuron concentrated in this triangle area called arcuate nucleus of hypothalamus. And research from the Richard Palmer's group published in Science in 2005, what they did is they generated a mouse model that they selectively express a toxin receptor called DTR in the AgRP neurons. by injection the DT toxin, they could selectively ablate AgRP neuron from the mice. And shown here is the picture that after DT injection, most of the AgRP neurons are gone. And then they did two interesting experiments. First, they injected DT toxin during the neonatal age of the mice. The mice can survive and grow up. At the age of 9 weeks old, they get two groups of mice and tested their feeding behavior. It shows that the AgRP DTR mice, which do not have AgRP neurons in the mice, show similar feeding behavior compared to their wild-type leader mates. And their fasting refeeding phenotype is also the same. But they did another interesting experiment that they injected the DT toxin during the adulthood of the 11 weeks old of mice. And you see here, it's clearly show that after the second injection of the DT toxin, the mice stop eating. They die because of hunger, because of the starvation. And this data based summarize that AgRP neurons are essential for feeding. And later it turns out really the activity of AgRP neuron that regulate feeding. There are several important examples in the field that indicating the firing activity of AgRP neurons are driving the feeding in satiated mice. I'm showing this study from Brad Lowell's group in the year 2011. What they did is they generated a mouse model that can selectively activate AgRP neurons by injecting a chemical called CNO. I think some of you may notice that the technique is called JET. And they did the experiment in the morning at 9 a.m. when the mice are well fed. If you work with mice, you know that the mouse usually eat during the dark cycle and they are full in the morning. But let's see how this mice showing. Okay, when they selectively activate AgRP neurons, the mice is eating, regardless it's full or not. But this wild type without AgRP neuron activity activated, he's not eating. Again, there's come another mice with firing activity activated in the AgRP neurons, he is eating. And again, there's wild type control mice, he's not eating. Okay, come another AgRP. I think that's good enough to indicate that if you activate AgRP neurons, it's gonna drive the animal start to eat, no matter it's full or not. Another piece of data from my own lab is, our own lab is, I use Optogenetics to specific activate AgRP neurons. And this can also drive the animal start eating, even they are well fed. You'll see here, sorry, okay. Before I turn on the laser, the mice is handling around, both of the two mice. But once I start the laser, these mice start eating like crazy, even though it is well fed. And as a control, we give the yellow light, which is not activate AgRP neurons, and these mice do not eat. So I think these two data just to show you that activation of AgRP neuron is essential for feeding behavior. And there's another important neuron paper from Brad Lowell's group. They actually did another experiment to get two different groups of mice and directly record the AgRP neuron firing activity from the AgRP neurons. And showing here, this vertical spike represents for one action potential. If you have more of this action potential, they indicate the more activated this neuron is. And they get the fed AgRP neuron recorded and found that if the AgRP neuron are fasted, they will show more firing activity in response to hunger. And this data indicate that fasting can turn on firing of the AgRP neurons. Now here we are dealing with a fundamental problem that we are dealing every day. That is, when we are hungry, we start to eat. And when we are full, we stop eating. And this is, so far we know that when the animals are well fed, they will maintain the AgRP neuron activity at a low level, and this will translate into a satiety signal to prevent the animal from eating. However, If you fasted the animal, and then this will drive the AgRP neuron firing like crazy. This will translate to a hunger signal to drive the animal start to eat. Our lab is interested in this fundamental phenotype, and we are trying to understand what is the molecular mechanism behind this and how these on and off AgRP neurons can coordinate feeding to meet the nutritional demands. For those who are not familiar with the electrophysiology, I use this slide to show how we do an experiment within the AgRP neurons. Typically, we get the mice in the morning and sec the mice to get the whole brain containing the hypothalamus. And then we section the mice containing the AgRP neurons, maintain them in a dish. Once we're ready for the electrophysiology, we will transfer one slice of the AgRP neurons in under microscope. As the AgRP neurons carry red signal, so we can directly see AgRP neurons and perform the electrophysiology under different models, like current model, voltage models. That's similar as others did. Our lab has been working on a few different circulation hormones that may regulate the feeding behavior. One circulation hormone I have been working is called Apolipoprotein I4, or ApoA4. What we found here is ApoA4 can directly inhibit the food intake in mice by inhibit AgRP neuron. The decreased firing activity of AgRP neuron is associated with decreased neuroactivity for its upstream glutamatergic neuron. On the other hand, we also found that another circulation hormone called heparin, fasting can increase the serum heparin so that will compete with insulin to bind for the insulin receptor on the AgRP neurons. By doing this, heparin will increase the firing activity of AgRP neurons so that will increase the food intake. But today, I'm not going to show you this detail for these two findings. I'm going to tell a story about the newly discovered hunger hormone called asparsin. And in this project, we hypothesized that asparsin promotes feeding while stimulating the AgRP neurons. This work is actually a collaboration with Dr. Intrinsic Chapter, who is a human genetic expert in Baylor College of Medicine. He is working with this rare disease called neonatal progeroid syndrome. This disease shown here, you will see that the first important phenotype is limb phenotype. One of his two patients has only around 10 BMI, the other is around 13. And this phenotype is because the two patients intake daily calorie are very low, almost a half of the normal female intake. Normal female usually intake around 2,000 calories. And Artur's work is trying to understand what is the genetic mechanism to cause this disease. And what he found is very similar. These two patients has a mutation in the same locus of the gene. And when he digged into the literature, it turns out that there are other 5 similar patients. They have the similar mutation in the same locus of the gene. So what is this gene? This gene is called FBN1 or fibrin1. And this gene will encode a protein precursor called fibrin1, which is important for the matrix, etc. matrix protein. And the mutation will cause loss of the C-terminal side products. So Artur named this product as asparsin. Now, based on their data, it shows that the cause of this MPS disease is because the patient does not have this asparsin in the plasma. As you will see the normal subjects that have certain amount of asparsin in the plasma, but the patient don't. On the other hand, they also found actually in the mouse and the rat, they can also detect asparsin in the plasma. This asparsin can increase upon fasting and will go down when animals are fed. At the end of the cell paper, to bring up a question, does asparagine regulate feeding? And this is the beginning of our collaboration. We then treat the mice by sub-Q injection of recombinant asparagine. As a control, we injected the mice with recombinant group GFP. You will see here, during a 24 hours food intake monitor, we found the mice received recombinant asparagine show significant high food intake during the 24 hours food intake. That is significantly larger than the GFP group mice. And then if we daily inject asparagine for 10 days, then the mice receiving asparagine will show significant increase of the fat mass compared to their control GFP group.
Yanlin, can I ask you a question?
Yes.
So these animals getting the asparagine, is there any change in their activity, or do you allow change in their activity, or are they in a restrained environment, or are you monitoring activity?
So there is no significant change of the activity between these two different groups. We just put them in their housing cage, and we didn't see any significant changes, except for they only intake more food daily. Yeah.
But other than the feeding behavior, they're not
other than that,
less
no.
active in any way?
No.
Okay.
Yes. All right. And then we also found there's a certain amount of detectable asparagine in the CSF, as is quite consistent with the plasma asparagine. When the animal are fasted, the CSF asparagine also goes up. This reminds us whether the peripheral aspirin can enter the brain. So we designed an experiment trying to confirm this. What we did here in this experiment is we add a histag in the asparagine protein. As a control, we have the GFP with histag, but GFP cannot enter the brain. They cannot go through the blood-brain barrier. And we found that there's a certain amount of histag exists in the CSF compared to the GFP group. More importantly, the nature mice, they will have endogenous asparagine in the plasma, so they will enter certain amounts to the CSF in the GFP group. But if we IP injection the histag asparagine, we can detect much more in the CSF. So these data indicate that the peripheral asparagine could enter the brain. Now we do another interesting experiment. We directly dump asparagine through ICV injection in the brain. So the mice can have better response to asparagine. And we found that upon injection of asparagine, the food intake in the dark phase significantly increased compared to the GFP injection. This data indicate that the central asparagine will promote the food intake in mice. Now the big question we want to ask is, what is the central mechanism for asparagine to promote feeding? Before I start this, I actually scanned a lot of different neuron types in the hypothalamus or the brain, which are reported to be important for feeding behavior. And I found that none of these neuron types, including dystrophic serotonin neurons, the SF1 neurons, PVH neurons, or dopamine neurons, they will respond to asparagine. However, I do find that about half of the asparagine neuron I recorded, they respond largely upon treated with asparagine. And this is quite striking to us, and we think probably this is the reason why the animal will promote feeding when we inject asparagine in them. Shown here is a typical trace of action potential firing from one single AgRP neurons. This is the trace that upon PUF treatment of asparagine, the action potential firing activity in the AgRP neuron shows significantly higher compared to the baseline without treatment of asparagine. Again, if you see more density of this action potential firing, means the neural activity is higher. And as a control, the GFP PUF treatment does not change the firing activity in the AgRP neurons. So this means asparagine could activate AgRP neurons.
Question. So in these current type experiments,
yes.
It looks like even at rest before the puffing of the asprosin,
Yeah.
that at least this cell is reasonably depolarized to whatever it is, minus 40 millivolts or so. Is that typical? Is that due to the ongoing firing, or do these cells ever sit quiet, and does their resting membrane potential get closer to minus 60 or 70 or not?
I think, yeah, you're right. So this typical trace is, I choose from many neurons we treat as person. And it just turns out this neuron, they show some hyperlyzed before we treatment. And their memory potential is slightly higher than the reported reference. But we do have some other amount of the neurons, they show low firing frequency, and their memory potential is also close to 53. So I think it's just variations, and we just pick up this one. Yeah, thank you. All right, and then we did another experiment, try to confirm whether asparsin directly activates AgRP neuron or it's through the upstream neurons, just like the ApoA4 experiment. And here in this condition, we treat the neurons with cocktail blockers, including TTX, bicuculin, and CNQX, APA5. These cocktail blockers will block the connectivity between AgRP neuron to its upstream neurons, and it's a common used method in the neuroscience field. And what we found is here, we can only see the memory potential changing because TTX already blocked the sodium channel. We don't see the action potential. But we do see that upon treatment of recombinant asparsin, the resting memory potential decreased, which indicate activation of the AgRP neurons. So by doing this, we confirm that asparsin directly activates AgRP neurons. More importantly, we want to confirm if AgRP neuron is the key role of asparsin targets. So we introduced the mouse model I mentioned before. We get the AgRP-Ablated mice and inject asparsin in these mice as well as the wild-type control mice. The wild-type control mice show significant increase of the food intake because they have AgRP neurons in them. However, the mice without AgRP neurons did not show increased food intake, which indicate that the asparsin mainly only target AgRP neurons to promote food intake. Now, the next big question we want to ask is, how can we mimic the MPS patient with a mouse model, as it's easier to work with a mouse model than directly work with the patient? What we do is we use the CRISPR technique to knock in one mutation from the patient I mentioned just now. And this mutation expressed in the mice will show significant lean phenotype, just like the human patient. You see here, this red line showing the MPS positive mice. After being 5 weeks age, they show significantly lower body weight compared to the wild-type mice. And these mice even do not respond to high-fat diet challenge. We put the two groups of mice within high-fat diet feeding for 8 weeks, and the MPS mice do not gain body weight like the wild-type control leader mice did. So this means with the MPS mutation, it's actually do not respond to the high-fat diet feeding. There are more phenotypes for these MPS positive mice. Their daily food intake show significantly reduced when they fed with adoliptin or they're challenged by faster refeeding. And more importantly, we found that the AgRP neuron firing activity within the MPS mice shows significantly reduced compared to the wild-type control leader mice. And after fasting, control mice, they will respond to the hunger and increase firing activity in AgRP neurons. However, the MPS mice failed to respond to the hunger signal. And then we did a rescue experiment because we know that the major problem of the human patients is because they lose asparsin in their plasma. And now we want to test if we give back asparsin in the mouse model, and what will the mouse perform. And the data shows here is when we get 3 groups of mice, the mice, wild-type mice are injected with GLP, and MPS mice inject with GLP. Last group of mice, we inject asparsin in these mice. We found that the wild-type control mice receive GFP show significantly higher food intake compared to the MPS mice receiving GFP. However, when these MPS mice receive asparagine treatment, they will recover their daily food intake. These measures indicate that asparagine could restore the phenotype of food intake in these MPS mice. And more importantly, asparagine injection mice also show restore effect in the firing activity of the AgRP neurons. Another important question is, we do notice that the DIO mice, which use high-fat diet, induce obesity, and OBOP mice, they also show significantly higher plasma asparagine. More importantly, the data from the human clinic indicate that the obese or human, they also have significantly higher asparagine in the plasma. So this gives us a hint that if we use some method to target asparagine, can we just develop an anti-obesity drug just by targeting asparagine? And that's the question, does asparagine antibody have anti-obesity effect? We then get a anti-asparagine mouse antibody. This antibody will specific to neutralize the active asparagine in the plasma and reduce the asparagine concentration in the plasma. By doing this, when I inject the antibody in the mice, this will significantly reduce the AgRP neuron firing activity. And more importantly, when the mice receive this anti-asparagine antibody, their daily food intake shows significantly decreased when they fed with adalimumab or challenged by faster refeeding. This data basically indicate that the antibody could basically reduce the food intake in the regular mice. Yes.
I presume when you conjugate the antibody that's function blocking, you also block the ability to transfer the blood-brain barrier of the complex. And so there's no asparagine that has preceded the injection of the antibody that's chronically maintained in CSF. Is it degraded, or how do you get rid of the asparagine that's already passed the blood-brain barrier?
That's a good question. So that's why when we do the experiment, it's actually 24 hours after we inject the antibody. And by that time, the already positive asparagine, it's already low. And also, then I think they are not good enough to respond to this hunger and also to their normal behavior, physiological behaviors.
But what is the central clearance mechanism for the elimination of asparagine?
Asparagine. I think itself in the paper supplemental field, asparagine, they have a half-wise of the life degradation around 12 hours. So I think naturally they were going to degrade or cleaved by the secretion.
Okay.
Yeah. But that's a good point. And then we want to try to repeat this experiment in the DIO mice. So we get the mice to feed them with high-fat diet and induce obesity. After the mice develop obesity, we repeat the experiment by injecting the antibody in the obesity mice. It shows that the asparagine antibody also decreased the food intake in the obesity mice. Upon 3 to 5 days daily injection of the antibody, their body weight significantly decreased in these mice, DIO mice receiving asparagine antibody. So this data indicate that it's probably a potential developed drug for the anti-obesity by targeting asparagine. Just to summarize what I have told you so far, first, we found asparagine is a hunger hormone which rises during starvation. Second, asparagine directly activates firing activity of AgRP neuron, which promote feeding. Third, lack of asparagine in MPS patient would lead to hypophagia, which can be rescued by asparagine administration. This is data from the MPS mouse model. And last, asparagine is elevated in the obese humans, and neutralized asparagine may carry therapeutic potential obesity treatment. All right, I think I already introduced you 3 extracellular secretion hormones that may regulate feeding. Now I'm going to move to another intrinsic mechanism of the AgRP neurons. Just to remind that I have shown you this picture a few minutes before. This is the study from Brad Loss group. And you see here, they still can maintain and get this data after the mice sacrificed. And this picture showing that after all this procedure and we start to record the neural activity from the AgRP neurons, it takes around 1 or 2 hours. Even after the mice, the neurons are removed from the circulation, they can still maintain this increased firing activity. So this gives us hint that probably there are some other intrinsic mechanism within AgRP neurons that will regulate the firing activity. And now I want to introduce another ion channel I am working on before I joined the lab. It's called small conductance calcium-activated potassium channel. This channel, when it's open, it can bring the potassium ion from intracellular to extracellular, which maintain the neuron resting membrane potential at a low level so that it can control the neural activity at a low level. But if you block the channel by its selective inhibitor, apamine, it will activate the neural activity. Interesting. I did an experiment trying to stand SK3 protein within the AgRP neurons. Here shows the image that at fed condition, the AgRP MPY GFP mice show highly colocalized with the SK3 staining, meaning that most of the AgRP neuron, they express SK3, but not other isoform of the SK channel. When I fasted the animal, and again, fasting will reduce significantly expression of this SK3 in the AgRP neuron. So that is very interesting because fasting can change an ion channel expression just within the AgRP neurons. At that time, there's another ELAB paper published from Dr. Stererson's group. They actually have the similar result as we did, but they focus on the mRNA expression of SK3 in the AgRP neurons. This data showing that SK3 mRNA level will significantly decreased when they are fasted, and other 3 isoforms of the SK channel do not express in the AgRP neurons. So based on all this, we hypothesize that the SK3-mediated current coordinate AgRP neural activity with the nutritional states to regulate the feeding behavior. Before the study, I generated a mouse model that can specifically delete SK3 in the mature AgRP neurons. Here is how I did. I get AgRP Kriya mice crossed with SK3 lox mice. By injecting the tamoxifen at the age of 8 weeks old, we can specifically delete SK3 from the mature AgRP neurons. And next, I need to validate if the deletion is successful or not. And shown here is a typical voltage current model recording the SK3 current from the fed and fasted AgRP neurons. Shown here, it's clearly shown that we can get a robust SK3 current within the AgRP neurons. And by applying the SK channel selectively blocker apamine, it can totally block this current, meaning this current is SK3 currents. As I mentioned just now, other isoform SK channel do not express in the AgRP, so we are confirmed that this current mediating is SK3 current. And in the fasted AgRP neurons, we cannot detect the SK3 currents. As another interesting validation is, in the knockout model, we failed to detect SK current, no matter in fed condition or fasted condition. This data validate that deletion of AgRP neuron is successful. Now, what will the neuron response if they lose SK3? The firing activity data showing that in the fed condition, the control AgRP neurons, they have SK3, they are firing at a low level. And when mice are fasted, SK3 expression is low, so that this will activate the firing activity in the AgRP neurons. As the SK3 deletion mouse model do not have SK3, so no matter in fed or fasted condition, their firing activity is always high. And with this phenotype, we wanted to know what will be the feeding behavior showing in this SK3 deletion mouse model. Here is the result. We basically get the two cohorts of the mice. One is control, the other is SK3 specific, delete SK3. We fed them with adalimumab and put them in the metabolic cage so that we can monitor 24 hours food intake. And the data showing here is that the AgRP neuron without SK3, this group of mice shows significantly higher food intake compared to their control leader mates. And more important is this data. If you look at the red circle here, the control mice, when they are fasted 24 hours and then do refeeding, as fasting can significantly remove the SK3 expression from the AgRP neurons. And the mice show significantly increased food intake during the refeeding. As SK3 is constantly removed from the SK3 deletion mice, so their refeeding is showing almost similar as their fed condition. So this data actually consistent with the firing frequency of AgRP neurons. In a fed condition, the deletion mouse do not have SK3. Their firing activity is already high, so they usually take higher amount of food intake compared in the fed condition compared to a fasted condition. Yes, go ahead.
If I recall correctly, SK3 is also associated with the myotonic muscular dystrophy.
Excuse me, what?
SK3 channel mutations are
Yes.
associated with myotonic muscular dystrophy.
How
do you reconcile your knockout of SK3, particularly with regard to lower food intake, which could be consistent with the dystrophy?
I think you're asking how can I make sure that deletion from the mouse model is only happen in the AgRP neurons, yes. Right? We do have a validation from different tissue from the deletion mouse, including the brain, the heart, the liver, and the muscle. And we found the deletion only happened within AgRP neurons because we are using an AgRP Cre ER mice. And this Cre activity only exists in the AgRP neurons, while not other tissues or the cell types. So by crossing with the AgRP Cre ER with the SK3 flox mice, they will only delete SK3 in the AgRP neurons while not affecting other tissue or cell types. So I think that's the key question. Okay. All right. Just to summarize the second part, we found that at fed condition, SK3 expression level is high, so this can keep the AgRP neuron at low firing level. It will prevent overeating. However, if the mice are fasted, the expression level of SK3 goes down. This will activate the firing activity of AgRP neurons so that it can promote feeding. And all right, just to summarize all I have told you today, I have been focused on a fascinating phenomenon that is the firing activity of AgRP neurons undergo the dramatic dynamic of daily-based response to the different nutritional states. And what we found here is we found ApoA4, which is a secretion hormone, can block the feeding behavior of the mice by block the AgRP upstream glutamatergic neuron activity. And we also found another secretion hormone, heparin, can stimulate the food intake by stimulate AgRP neuron activity. A newly discovered hunger hormone, asprosin, can also stimulate AgRP neuron and then promote feeding. There's another intrinsic mechanism of SK3, which expressed highly in the fed AgRP neurons and expressed low in the fasted AgRP neurons. The SK3 control the response of on and off switch of firing activity of the AgRP neurons. And for yes,
before you leave that,
okay,
the back segment,
yes,
two quick questions. One,
sure.
so, all right, first of all, just a simple technical question.
Mhm.
How long into the fasting does it take, do you see that response and the change in firing rate? How long was it when you actually collected the data? I forgot.
Yeah, that's a good question. We actually did a lot of experiment, try to understand the timeline for fasting. In this type of experiment, we usually fast the animal for 24 hours. But I did a lot of recording by fasting animal for 1 hour, 2 hours, 4 hours, and 3 hours, as I can detect the earliest change of SK3 in the electrophysiology level is 4 hours fasting during daytime. During nighttime, we try to fast the animal starting from 5 p.m. to 7 p.m. It even change larger, but it's not totally remove to the significant reduction of the SK3.
Okay. Do you have enough data points to have a timeline that you could say something about the function? That is the firing rate a linear relationship to the change in these circulating levels, or is there some kind of threshold where it takes off?
Yeah, that's a good question. We do have the data, but we didn't put it into such kinds of figure. And we also try to collect the sample from the hypothalamus of including the AgRP neurons by doing the qPCR to indicate the changes expression level, SK3. And we didn't put in the paper, but we do have such kinds of data showing that the SK3 as a time point of decreased expression level at about like 6 hours fasting, there could be achieve a significant reduction upon the. But typically, as it's convenient to do the experiment, we will remove the food in the morning when we come to the lab, and then we start to record in the next day.
Okay. And then my next question is just, so maybe you're going to get to this. If you do, that's fine.
Mm-hmm.
So what you've shown here at this point, this conclusion, is kind of a master regulator neuron of all these signals converging on these agouti neurons. And I forgot early on you showed us pictures of them and how many. I don't remember how many.
There's a lot actually.
That's okay. You don't need to go back.
Yeah.
But my point being, you have a very interesting and complex behavior, a whole behavioral repertoire in terms of eating here. And you have a group of neurons that I think are a relatively small subset of the neurons in the nucleus. So it's a wonderful system for having a convergence of signals that gives you a relatively simple output circuit that controls this whole downstream behavioral repertoire. Are the numbers of these cells quite small? That was my impression. Is it just a couple hundred or a couple thousand neurons? Or what?
A few years ago, my mentor, Dr. Xu, count. It's around 2,000 within the hypothalamus, 2,000 AgRP neurons.
Okay.
Yeah.
All right. Good.
Okay. I have a few more minutes. I want to discuss a little bit about my future plan. So upon the road of Asperson, I think you will remember there's an important question is, why only half of the AgRP neurons respond to Asperson? I think that's related to the receptors. So far, we're trying hard to discover the Asperson receptor in the AgRP neurons. And we do have some clue, and we also have some data. We have a potential target. We get the genetic deletion mouse model. It works good. We can repeat the phenotypes. But as this is a collaboration project, and it's going to be finished within a few months, so I guess my mentor do not want me to show the preliminary data here, but I can tell you that we are trying hard to get the Asperson receptor discovered in the AgRP neurons. It's going to be published soon, hopefully. And the second important question is, can Asperson regulate SK3 in AgRP neurons? I think that's why I put these two stories together, because I already got some preliminary data. It's easy to test upon treatment of Asperson how SK3 changes. And the data showing out when I pop Asperson in the AgRP neurons, it does not change SK3 current by acute treatment. However, we do find a significant change in the MPS mice. Remember, the MPS mice do not have high Asperson in their plasma and also their brain. And this is indicating that something happened for the Asperson to regulate the expression level of SK3, but it's probably something that relates to the long time expression level. And if we can successfully find the Asperson receptor, then probably it can be easier for us to detect how the expression level changes. Okay, and I think I will spend more time to dig out this by just trying to target SK3. And last, I think it's also important is how to understand the structure of the asparagine protein. And I can tell you about 4 years ago, Dr. Chopra is already collaborated with some other experts in the structure. They are trying hard to get some mutations and try to understand what is the structure of asparagine protein. I helped them to test about maybe 60 different mutations from different sides of this protein. Some of them found they mutate at certain site, they lose the function of asparagine, but they're still trying hard to finish the whole structure. I'm not sure, maybe 1 or 2 years, try to discover the structure. And if I have the chance, I will still collaborate with them, try to understand the mutation sites and take part in the job. So with that all, I want to thank my mentor, Dr. Xu. He is a very good scientist and also helpful mentor. He trained me for a short time, I guess 5 years. I spent only 5 years with him, but I learned a lot. He wrote around 20 different proposals every year to get funding. He also trained every postdoctor in the lab to write at least 2 every year proposals. Try to get everybody has their own fellowship or grant. And I think it's useful. And I also want to thank our collaborator, Dr. Chopra. He's the guy that provides the resource of asparagine and also the animal model for us to perform the experiment. I want to especially point out that we have 2 Virginia Tech alumni. Dr. Chen Liang is from, I think, Dr. Norcock group. He just joined a lab last October, so he's not in the picture. And Dr. Pingwen Xu moved to University of Illinois at Chicago last year, so he's also not in this picture. But these 2 students are outstanding students from Virginia Tech. That's why I have very interested in joining here, because I think I can get support from the students here, and also I can collaborate with them or train them to do the research, and we can become successful together. And with that all, I want to thank everybody's patience, and if you have any questions, I would be happy to answer. Thanks.
Thank you. Okay, for questions, comments?
You mentioned in obese people, asparagine has elevated plasma
Yes.
levels. Yeah. Does that precede the obesity, do you know?
It's actually the volunteers from Baylor Clinic. I don't have very detailed information, but it's just Baylor usually have some kinds of program that if you volunteer to come to the clinic and get some blood draw, the background of the volunteer is actually I don't have access to the details, but definitely their BMI is quite high compared to the normal subjects. Other patients, whether they have like binge eating to induce obesity or some other information, sorry, I don't have. Yeah. Yes.
On that perspective, way might be difficult, but it might be also interesting to look at whether asparagine changes with weight loss too.
Weight loss. Yeah, that's a good point. Yeah, so the patient, they do have a very lean phenotype. They don't have too much fat. Yeah, that's a good point. Yes, go ahead.
The OB mice,
Mm-hmm.
they are leptin deficient. What is the interplay between asparagine and leptin?
That's a good question. I guess that's probably a question that raised up by the reviewer when we submit the Nature Medicine paper. I think the situation here is the independent mechanisms. The leptin resistance of the opioid mice, they are not responsive to leptin, but leptin is not the only factor that affected the body weight and food intake. Asparagine could be another important regulators. So I think there are 2 different independent mechanisms. Yeah.
So do you know anything about the proteasomal degradation of the SK3 channel or the transcriptional regulation? It's a really interesting mechanism. Seeing how daily fluctuations in feeding behavior can lead to changes in channel expression with a functional consequence. And then relatedly, how is the regulation of SK3 in other tissues besides AgRP neurons? So the AgRP neurons obviously are responsible for the feeding behavior, but SK3 must have a role in other cell types as well.
Yeah, you raised a few questions. Let me answer your first question. So your first question is whether there's a clue that how asparsin changes according to the feeding state. To be honest, I don't have the answer for this question because so far we don't know how asparsin cleaved from the body and how the
SK3.
Oh, SK3. Oh, yeah, yeah, that's a good question. SK3, unfortunately, the only factor we know is FOXO1, as I mentioned in the Cell Reports paper. So FOXO1 can change the expression level of SK3, and by stimulate FOXO1, they will be able to coordinate the change of SK3. And your second question, yes, we also have some other projects that relate to SK3. Like I have an ongoing project, the SK3 within the serotonin neurons, and we found that it's an important regulate for the binge eating. Unfortunately, maybe some of you are interested for the binge eating today, but I don't have time to explain, but I will be happy to discuss with you afterwards. And we found that SK3 shows the deletion of SK3 in the serotonin neurons will significantly reduce the binge eating behavior. And we're trying to publish that part of the paper, and probably we'll see you soon. I guess my answer touched your point. Yes.
Just curious, when you use the genetic method to directly activate the AgRP neurons,
yeah.
the mouse start eating behavior immediately. How long it takes for the feeding behavior to disappear after use of the gene stimulation?
Okay, so sorry, do you mean how long do we need to wait until the expression of the optogenetics, or how long after turn on the laser? Turn on the laser. So I mentioned in the video, it's played by 3 times faster. So typically it's like about 30 to 45 seconds delay when I turn on the laser. The mice start to search the food and bite.
So is there any way you can monitor the asparsin level in plasma before and after you using the genetic stimulation?
That's a good point. We never thought to do that type of experiment, but as we don't have a real-time monitor for the asparsin in the body, so what we can do probably just stimulate the mice for 10 minutes or 1 minute, and then second the mice just to get the blood and test. But it's not like the glucose. Now we will have the glucose monitor to real-time read the glucose changes. For asparsin, it's a long way to get that kind of
curious, does the overactive of the AgRP neurons gonna cause the asparsin level
changes?
Yeah.
Well, I don't know the answer, but I guess it's gonna be hard because originally asparsin produced in the fat tissue.
Okay.
Yeah.
I just want to ask you one other question. I've
Yes.
asked you a few, but can you tie this back to the serotonergic modulation of the dopaminergic system, the ascending reward system, and how this feeds into this whole thing? Are you going to talk about that tomorrow, perhaps, or is there anything else you want to share with us?
I will talk more about tomorrow about this. Yeah. It's actually, I think I still have a few more minutes. I can share you a story. So the binge eating, actually, when we published the paper, we found actually not that many people accepted that we are performing the experiment in the mice. They thought you cannot mimic a real binge eating in human. That's why when we submitted for the grant, I remember in the year 2014, NIDDK clearly indicate that they won't accept such kinds of proposal. I don't know the reason, but definitely. And also when we submitted the manuscript, the reviewers also point out that you use a mouse model to mimic a human psychiatry behavior. It's gonna not be that perfect. But so far, this is the only mouse model we can use to study these kinds of disease. I know some experts here, they study the alcohol addiction, and we also use almost the similar mouse model. But yeah, still, there's no fantastic mouse model to study for the human disease.
Yeah.
Yes.
I was wondering, when the asparsin appears in evolution, if it's a mammal thing and if it's also conserved
Yeah, so far we only did the rodents model, we did the rat and the mouse. Not sure, I'm not familiar with other animal models, but I know someone is using the rabbit or the dog, but we never tested it.
Invertebrates,
No.
you can find the system and these neurons are conserved during evolution.
If I guess, they probably share the similar mechanism, but I don't have the data.
But there are no non-mammalian vertebrate systems that have been studied yet. I gather it's all mammals, right?
Yeah. Yeah. Right. Thank you.
Okay, if no more questions, please join me in thanking gentlemen for a great session.
Thanks a lot. Thank you.